to turn now Dr. Kirill Martineov with the Martineov Lab with the University of Florida is one of our scientific advisors as well on the Bow Foundation, and he's going to help us moderate the rest of this morning's session. So we'll invite you to kick us off.
All right, let's get the science continuing going. We have a very exciting, set of programs. We have 10 talks on the cutting edge of science from the leaders in this field. And what's going to be important is to keep the timing tight. So I'm going to be sitting right here, listening very intently to every word that you have to say. But when your time is up, you're going to see me stand up, and that is a signal that that's probably time to wrap up. Anyways, with this, it is my pleasure to invite Terry Hebert here from McGill University in Canada. And Terry has been studying G proteins for quite some time. And now he's in this clinical space of the GO mutations and G beta mutations. And the title of his presentation is the alpha and beta gamma of GNO signaling.
Thank you very much, Kirill. I look forward to you not standing up. I wanted to thank the Bow Foundation, first of all, and for Emily for reaching out to me. And I think that we have a lot to talk to each other about. During the break we were talking about alpha and beta gamma. Those of us who study G proteins don't ever think of them as separate proteins, right? We think of them as a unit. So you've been talking about the GNAO alpha, and you'll see from your families, you'll probably recognize your mutations, and a lot of it affects GTP binding and the activation of adenyl cyclase, for example, downstream effectors. But there's a bunch of them that cluster in an area that are important for interactions with their family members, the beta and gamma complex, right? And you're going to see all of the symptomologies that you've been talking about, the movement disorders, the epilepsy, even the gastrointestinal problems. Those come back in mutations of the protein that I study, the GNB1. So I'm going to switch over from talking about the alpha subunit to talking about the beta gamma subunit. But always keep in mind that I think they work as a unit, and I think that the beta gamma subunits are really critical for supporting and in some ways regulating the functions of the alpha subunits and vice versa. So maybe patients with GNB1 mutations have compromised G alpha function. Maybe patients with GNAO1 mutations have compromised G beta function. So I'm going to talk a bit about the beta gamma. Kirill sits in really between both worlds. He works on this disease, GNAO1. He also works on GNB1. And for a long time, he's been showing things that we've been seeing too, is that G beta gamma subunits, beyond their canonical effectors at the cell surface, they're found all over the cell, and they're doing different things in different places, right? So G beta gammas, we've shown over the course of many years, is that they're involved in building GPCR signaling complexes, right? They're involved in getting the partners to talk to each other as they go through this journey from genes being turned into proteins and then moving those proteins out to the cell surface where they do their work. So what I'm going to tell you about maybe is how G beta gamma subunits do things beyond these established canonical roles that are important for trafficking. And I'll tell you a story about how they might even be important in the nucleus of different cells, acting as transcriptional regulators. So the GNB1 encephalopathy, very many of the same symptoms, right? Epilepsy, abnormal muscle tone, developmental delay, all of the things we've just heard about, right? And so if we look across the space, so this is a busy slide. I'm sorry, I have a lot of busy slides. The gold is the alpha subunit, right? So that's the one we've been talking about. The green is GNB1 or G beta, and then this silver one here is the obligate partner of the beta subunit, which is called G alpha. You can see that there's a bunch of mutations in G alpha or in G beta that affect its function. The blue ones here are ones that are germline, that children are born with or inherit from their parents. The red ones are somatic, and sometimes they occur spontaneously in adults, right? So in the germline ones, we see symptoms just like GNAO1. And in the somatic ones, we sometimes see their effects in cancers later in life. So the other thing we almost never talk about in the G beta, G alpha world is the gamma, right? The gammas are the ones that are most different in terms of physical structure. So we think that like thinking about what the gammas are doing is going to be a critical area of work going forward, right? So, but I'm not going to do that today. I'm going to try to keep it relatively straightforward. The other thing that I will torture you with today is that I'm going to talk about aspects of G beta gamma signaling that aren't necessarily related to the nervous system, right? So I'm going to drag you into the world of the heart, where we identified a lot of the things that we see with G beta gamma. This kind of summarizes a lot of work that was done to show that the G beta gamma subunits themselves play a huge role in heart failure, and you can treat animals with angiotensin, which is a driver of something called cardiac fibrosis, and you can see that those animals will get fibrotic hearts, stiff hearts that don't function as well. And the cool thing that was found a few years ago is that when you drive hearts into failure, you see a lot of fibrosis, all of this pink here. But if you treat with a beta gamma inhibitor, you prevent that fibrosis from developing. So we were thinking about how to understand the role of GNB1 in cardiac fibrosis. And the way we do that in our world is to knock down the expression of the gene using something called small interfering RNA. And what we saw was a surprise to us is that when you treat with angiotensin, you get a fairly significant fibrotic response at 24 hours. But when you knock down G beta, that response is actually upregulated. So there's a lot more genes that get turned on in the absence of G beta 1 than in the presence of G beta 1, suggesting that GNB1 or G beta 1 is acting as a brake here, right? It alters the fibrotic response. And maybe it's a way for the cell to determine whether or not we want a fibrotic response, right? It says no, 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 and then eventually it says yes. You could see the same thing at the protein level. When you knock down G beta, you see a lot more proteins being upregulated in the fibrotic in the heart or in cardiac fibroblasts, and you see a lot less proteins being turned off, right? So it looks like at both the gene and protein level, G beta 1 is acting as a brake. So this is where we started to get into this rabbit hole of G beta signaling, which kind of Kirill has been important in kind of defining the extent of how far down the rabbit hole goes, but everywhere you look in the cell, everywhere, you can find G beta gamma subunits. And the weirdest thing to us was we started to see them in the nucleus. And one of my students, a very long time ago, came into my office crying because she thought the experiment didn't work, because it showed that there's an interaction between G beta gamma and a nuclear protein. That's not right. And I said, well, maybe it's a control gone horribly wrong, or maybe it's something new. And so eventually we found, and others have found, like G beta gammas interact with a lot of different transcriptional regulators, right? So they're controlling gene expression in some way. And a few years ago, we found by accident that one of the things that G beta gamma interacts with is RNA polymerase, which is the main driver of the formation of RNA, of messenger RNAs, right? So that was an odd finding. And so we thought we would trace that down a bit and try to figure that out. And so we started to kind of do proteomic studies to kind of find the interactors for G beta gamma. And one of the things that we were surprised by was that there were a lot of things that are associated with the nucleus in that, right? Including things that shuttle other proteins into and out of the nucleus. So we thought, okay, here's a potential mechanism for how G beta gamma gets into the nucleus. What's it doing there? And then we started to ask, well, what is it doing there in response to canonical receptor stimulation? It turns out that, like, the interaction that we found with RNA polymerase is increased when you treat cells with angiotensin, right? You can see that 3 times or summarized here. So G beta gamma subunits, thought to be a cytosolic protein that lived mostly at the membrane, is somehow getting into the nucleus and interacting with a regulator of RNA production. If you block entry into the nucleus, you block that interaction. That was interesting too, right? You can block the uptake. And then the most exciting thing at the time for us was that you can actually pull down a protein from the nucleus and look for which genes it's sitting on. And so when we do that, where we either use to control siRNA or knock down G beta, you can see that, so under control conditions, a lot of genes, so this is all of the genes in the human genome, there's RNA polymerase sitting along the length of all of these genes. If you hit those cells with angiotensin, the number goes up. If you knock down G beta, you increase that number dramatically from control, and when you treat those cells with angiotensin, you get a lot more genes that have RNA pol 2 sitting on them. So we did that with G beta as well, and you can see that there's a cluster of genes where there's G beta actually interacting with the genes, right? And so this is all the genes. This is a global picture, but we could see that when we looked at individual genes as well, right? So G proteins, G beta gamma subunits are interacting with the systems that regulate the production of other genes and other proteins, right? So where did we go next? We wanted to figure out how G beta gamma got into the nucleus. We wanted to figure out if it interacted with some of its canonical partners there and other transcriptional factors. And we wanted to understand how to build that network outward. I won't tell you about any of that today, but we started thinking about it, and this is where I first heard of the Bow Foundation, is how to connect these findings back to G beta gamma, G alpha signaling, right? So we could show that all of the G proteins in the cells we were looking at interact with G beta in our screens. But weirdly, we also started to see, and this is another Carroll connection, because he was very interested in these proteins as well, a family of proteins called KCTD. So the idea here was that we were interested as a potential therapeutic in being able to toggle the levels of G beta gamma. So we wanted to understand how those levels were controlled. And it's been known for a while that a lot of these KCTD proteins are involved in controlling the levels of other proteins. So I won't torture you with this, but we see a bunch of different KCTDs, and we validated that with classic biochemical methods. But then we wanted to kind of look at what KCTD was doing to G beta, and it took 10 years for this study to come out. I have to say it took 10 years for the study with the gene expression to come out, but we finally got a structure of this large complex involving 5 KCTD proteins, 5 culins, so these sort of adapters that target proteins for destruction, and then 5 G alpha or 5 G beta gamma subunits, right? And that's probably better shown in this little movie if it runs, and you get a sense of how intricate these machines are, right? This is a machine that is purpose built to control the levels of G beta gamma in a cell. And you can see that's a pretty cool little thing. Yeah, I guess pretty cool to me anyway. So what happens when you tinker with that interaction? So here we're actually looking at the formation of beta and gamma, and by using a technique called biomolecular fluorescence complementation. And you can see that when you knock down KCTD5, you increase the amount of G beta gamma in the cell. And you can rescue that by putting KCT back. You can do that in a knockout line here. Now we've knocked out KCTD5, and you can see that you get a lot more where is my guy here? You get a lot more G beta gamma being made, right? And then you can rescue that by putting back the wild type KCTD5, but not with 3 mutants that interfere with the beta gamma interaction. And the other thing we showed that in that knockout lines, you reduce the ubiquitination of proteins associated with beta gamma, right? Without KCD5, there's no ubiquitination. So it's controlling the levels of G beta gamma. So what are the implications of that? So then we knocked out GNB1, right? And I only want you to focus on one thing, right? So the red means something goes up, the blue means something goes down. Look what happens to G alpha and GNAO1 when you knock down G beta, right? It goes up dramatically in one cell line, okay? And that it was also modulated by knocking down different KCTD5s. So there's some relationship between the function, the levels of G beta, and the function and levels of other G protein partners that they interact with. It's all connected. And the last little thing was we also thought one of the things that we saw when we knocked out G beta gamma was the activity of a bunch of proteins called SMARCAs that are actually important in controlling access to genes, right? They allow genes to be accessed by the transcriptional machinery. That, when we knock out G beta, the activity of the SMARCA system goes up. And we can see that again in two different types of cells, that SMARCA4 is interacting with G beta. And so when we come back to that, when we knock down, this is now back in a different cell type, and you can see that, again, there are effects when you compare control to knocking down GNB1 in a primary cell, but they're different than the last cells, right? So it's cell specific, it's G protein specific, and we thought this is, again, another way of showing that there's no way to separate out the effects of G beta gamma on G alpha, right? G beta gamma is doing things even independently of its role as a partner for G beta gamma by controlling the levels of G alphas in the cell. Same story with G betas and G gammas. So where do we go from here? We have a bunch of big questions that we still want to answer. We have all of these different GNB1 mutations. We want to test the effects of modulating their levels or the levels of KCD proteins on shaping the signaling landscape. That's why I think a lot of the effects that you see in your children are both common and distinct. It depends when the mutation happened, it depends where the mutation happens, and it depends which cell you actually look at, right? So we want to understand the broader impact of the 50 or so mutations in GNB1. We want to be able to target those things, right? So the last talk was really interesting because we were approached by a company who wants to build solutions one patient at a time to make a G beta gamma ASO that would actually rescue function by skipping, by exon skipping. We want to figure out the relationship between the G alpha subunits and mutations in GNB1, and now more recently mutations in G beta 2. And we're sort of switching to more a human relevant system by now making iPSCs, so induced pluripotent stem cells, from patients who have different forms of GNB1 mutations. So Carol didn't stand up. I did well. I have an amazing group. 10 years of work from these two guys to get us to the story in the nucleus, and 10 years of work from Darlene, Robert, and Dominic to get us to. I mean, I have a picture of Dominic with his small child on his back, and he's been with me so long that now he's on the back of his not so small child now. And then the three other people to thank were Jason Tanny, my colleague at McGill, Paul Clark, another colleague at McGill, and Gil Privé, who we did the structure with at the University of Toronto. And again, I thank you. The engagement of families, putting all the stakeholders in the room together is the best way to do this. So I'm quite happy to have been able to speak to you. Thank you.
Well, thank you very much, Terry, for this wonderful talk. We've got time for questions.
Thank you, Carol. Thank you, Terry, for this great presentation. I was just wondering, so you have a link for G beta proteins to SMARCA. So have you also done the reverse, like knock down G alpha and see whether G beta levels go up or down?
Yeah, I'm going to reveal my prejudices here. I'm a G beta gamma person, so I haven't tried to knock down the G alpha. We've been thinking of G beta as a kind of a central regulator of the whole signaling networks of the landscape for a while, but there's no way to avoid the relevance of your question, right? So we haven't tried yet to knock down any of the alpha subunits.
So you talked a little bit about how mutations in the system affect the heart. Do we know anything about it, how it affects the gut?
No. I think at the level of the basic biology, which we're kind of doing, I don't think we've done that, right? And I think that the window into that now is, in the last few years, my lab has invested a lot of time and treasure in being able to build a pipeline from patient to iPSCs to whatever cell we want. So we're not a gut lab, so we've been focusing on making cardiac cells, and more recently on cortical and dopaminergic neurons, and then even more recently, because grant reviewers told us to do this, to make kidney organoids, but we haven't done gut organoids. But just across the city from me is the guy that's going to get that question asked. So yes, we'd like to do it, but there's only so many things to focus on.
How about you? Any other questions? No, thank you so much, Terry.
Thank you.
Yeah.